Dormancy is a way of plants to ensure their survival and perpetuation, but seeds cannot just remain dormant forever. There is an expiration to the viability of seeds, and if they fail to germinate, then the chances to produce another generation of the same plant species are lost. At some point, the seeds need to germinate. Nature has various ways of breaking dormancy in seeds to encourage germination. In this lecture, we will talk about nature's way of breaking dormancy. In particular, we will look at how seed dormancy is broken down by cycles of freeze-thawing of the soil, animals, forest fires or smoke, and by plant hormones. Let's begin with the natural cycles of freezing and thawing of the soil. Temperate plants that produce fruits during the cold season tend to deposit seeds in the soil seed bank when temperatures are very low. As the season transitions from winter to spring, we can expect temperature fluctuations. These temperature fluctuations, combined with harsh winds and rains, can soften the tough seed coat by gradual expansion and contraction of cells. This constant expansion and contraction eventually disrupts the testa at weak points, allowing the seed to imbibe water and take in oxygen, which are necessary for germination. The cycle of freezing and thawing of seeds in the soil mimics stratification, which is a germination requirement for seeds of crops such as hawthorn, wild roses, apples, and hops. Animals also aid in breaking dormancy in seeds. Dormancy can be broken down by passing through an animal's digestive tract after ingestion. The acids in the digestive tract of the animals scarifies and weakens the seed coat, allowing the seeds to germinate once the animal excretes the seed. Cassowaries are a good example of nature's way to break dormancy. Cassowaries are large birds in Australia that forages on large native fruits on the forest floor. The bird swallows the fruit whole. After carrying the seeds in their guts for a while, they drop them elsewhere in their poo, as you can see here. This process is very beneficial for seeds for many reasons. Number one, the seeds are scarified by the acids in the bird's gut, essentially breaking the dormancy of the seed so it can germinate. Number two, because the birds drop the seeds in their poo, the seedling that will emerge from the seeds would be far from the mother plant, therefore far from competition. And three, the poo of the bird provides a very fertile environment for the seedlings that will emerge from the seeds. The emus, as pictured here, are also big birds that feed on the fruits of snotty gobble, which is the shrub pictured in the right. Upon ingestion by an emu, the seed of snotty gobble is scarified in the emu's digestive tract and gets coated by the emu's gut flora and other chemical substances that can signal germination for the seed. Aside from birds, we also have nature's helpers in the form of ants and bees. In Australia, there are species of ants that feed on the stalk that attaches seeds of the acacia plant to the fruit pods, and that is the orange thing that is encircled here in red. The stalks containing the seeds are usually carried by ants to their nests underground. In most cases, the ants carry these seeds to a distance of 180 meters. The seeds are brought underground to be fed to the ant larvae. The burrowing by the ants loosens and aerates the soil, allowing water and air to penetrate in the chambers. This moist and aerated environment underground breaks the dormancy of the acacia seeds, allowing them to germinate. Foraging by bees is another example of how animals can aid in breaking seed dormancy. These are Cadaga eucalypt fruits. Each capsule that you see here contains sticky resins that keeps the seed inside. If the seeds are not manually removed from the capsules with the sticky resins, they will not be able to germinate. Now there is a particular species of bee known as Trigona carbonaria that collects the resins from inside the fruits and use it to build its nest. This is the Trigona carbonaria bee. By foraging the sticky resin inside the capsule, the bee is actually freeing the seeds from the resins and helping them gain access to water and oxygen that are necessary for germination. Fire and smoke are also natural means of breaking dormancy in seeds. Heat caused by the fire or smoke caused by the combustion of organic matter are important in a number of plant ecosystems. Some plants actually require fire for the seeds to germinate. Panel A is a satellite image of African wildfires in 2002, while Panel B is an image of Australian bushfires in 2013. These spontaneous fires, which are indicated by the red spots that you see here and also here, are necessary requirements for healthy ecosystems in the region. How does fires aid in breaking seed dormancy? Let's take, for example, the case of a coniferous plant known as Banksia serrata. Banksia serrata seeds are kept inside fruits known as follicles, and that is the structure that you see here. This is an example of a mature fruit, and this is an example of an immature fruit of Banksia serrata. These fruits are sealed off with a resin that can only be physically melted by the heat caused by bushfires. Without bushfires, banksia are able to keep their seeds in the fruits and they can remain there for many, many years. The same is true for lodgepole pine. The cones of the lodgepole pine, as shown in panels A and B, are sealed off with a resin. These cones will only open when exposed to very high temperature, such as those produced in wildfires. Without the natural fires that spontaneously occur, these cones will not open to release new seeds. Panel C shows young lodgepole pine saplings in Yellowstone National Park a few months following a forest fire in 1998 that burned 200-year-old trees. For some plant species, it's not the heat from the fire, but the chemicals that are found in the smoke that are the key to breaking dormancy. Some species of wildflowers, shrubs, and annual plants, such as Calatrix lecionoltii and Lecionoltia biloba, require the chemical signals from smoke and charred plant matter to break seed dormancy. Smoke has a chemical component known as carikin that has been identified to promote germination. Carikins belong to a class of molecules known as butenolides. It is a product of combustion of cellulose together with organic compounds found in plant tissues. Panel A illustrates the core structure of carikin. To date, several forms of carikins have been identified including carikin 1, 2, 3, and 4. Among them, carikin 1 has been reported to induce the expression of two key gibberellin biosynthesis genes. It is proposed that carikins work by interacting with ABA-GA signaling networks. If we look closely at the chemical structure of carikin here in the left, you would notice that it shares similar components as the plant hormone strigolactone, the chemical structure of which is here in the right. These two rings here are reminiscent of the chemical structure of carikin. Strigolactone or strigol was first isolated from the root exudates of cotton and has been widely used as striga-trap. Strigol can stimulate germination of striga weeds even in the absence of host plants. These are striga weeds. Striga is an obligate parasite that feeds off of the host plant by siphoning nutrients from the xylem and phloem of the host to itself. This is a field of striga plants in a cornfield in Africa. You can clearly see that there are actually more striga than corn. Striga produces 90,000 to 500,000 seeds per plant that can stay in the soil seed bank for more than 10 years. Seeds of striga are highly dormant, but can be stimulated to germinate by signaling molecules known as strigolactones that are released by the host plants like sorghum or corn. Nature's way of breaking dormancy may take a long time to complete, but the longer time requirement to break dormancy in seeds actually expand the range of time over which seeds can germinate. Most modern crop species have undergone domestication and selection for rapid and uniform seed germination. Both exogenous and endogenous seed dormancy can be overcome by natural means. In summary, plants in their natural environment depend on nature to overcome seed dormancy towards the renewal of plant life. Some of nature's way of breaking dormancy includes the natural freeze-thawing of the soil, animal-aided breaking of dormancy, microorganism activity, forest fires and smoke, and plant hormones. Domestication of plant species has selected for rapid and uniform germination of crops. Dormancy in cultivated crops can be overcome by mimicking nature's way of breaking seed dormancy.